This will serve as a relatively short introduction to the final exam. The things that you need to worry about are listed on Canvas, and I'm just going to go over a few things to remind you of them. The final exam itself will be similar in nature to most of the exams we took. For this purpose, it is 60 points in terms of multiple choice, taken online, has to be completed sometime on Wednesday, May 18th. The take-home portion is 40 points. There were 5 questions worth 8 points apiece. That is available now through Wednesday, May 18th. It has to be turned in just like the other one before midnight on that day. The things to remember for the comprehensive exam: if you've been successful on lecture exams in the past, do the same thing, right? Things that have helped before have been taking a look at the lecture notes. We have videos for some of the lectures, right, that are still online. If you can't find them, if they look like they're collapsed, then just push the little triangle and that'll open up all the list of things. Not sure you have time to listen to all the lectures that are posted online at this point in time, but certainly you could go back and review things. The group problems have been important. The lecture notes have been important, and also there are learning objectives for the comprehensive exam as well. So make sure you look at those. There's a set of student answers posted. As you read through them, if you have a better answer, then that probably is a better answer. I posted the document as a Word file so you could edit it. One of the things that's important is reference what's not on the comprehensive final, so you can just make sure you don't study that. You'll be smarter if you do, but you won't be as prepared. You only have a limited amount of time between now and then, so make sure to maximize your studying time for that. I would review all the notes from class from the first day to the last day. I would do that as many times as I could. I would certainly go over the group problems. I'm not sure there's enough time at this point to write every one of them out, but you might want to, or at least go over them slowly. You can certainly expect some of them to be on the exam in some form. There is a systems comparison chart. That systems comparison chart kind of looks at the different systems and looks at what's the relationship between them. And so it takes the systems we spent a lot of time on, the nervous, the endocrine, the respiratory, the cardiovascular, and the renal, kind of tried to link them together. It's a little superficial, but it kind of gives you a nice overview. One of the things is you should try to anticipate possible questions. The group problems, some of the questions, we can't change much, right? If I decided to ask you about, say, the differences between the autonomic nervous systems and the somatic nervous system. So, draw a schematic of that. How much can I really change that for a question, right? But other things you certainly can change significantly. And so you'll see some things that look familiar and some things that look different. And when you get the choice, pick the one you can get the most points on, right? So, practice or at least think about it. For the comprehensive final exam, I moved it near the modules, the top of it, so it's easy to find. There's a list of formulas and equations you might need for the exam. And that starts way back at Fick's law, or the number of dissociable particles to calculate osmolarity times the molarity, all the way through the VA, VE, cardiac output, Q stuff that we had on the last exam. Look at the tips for preparing for the comprehensive exam. Again, look at what's not on the comprehensive exam, the learning objectives, the student key for that, the comparison chart. There's also a famous people um, one. So if you want to kind of get the people in mind, that helps. And some of that comes from Elizabeth's lecture notes. So if you kind of scroll down, it's right at the edge of the difference between the lab and lecture notes on Canvas. So once the had all the lecture notes, then it starts to be lab notes, and hers and the other students' notes are right there. So that's kind of, things to look at. We'll look at a few other things though, and kind of go over a couple of other things. So here's what's not on the comprehensive exam. Again, I think that this is something you should pay attention to make sure you don't study things that you would think would be on the exam but aren't. And that's mostly because they're not on the learning objectives. And so if they're not on the learning objectives, they're not on the exam, right? So some of the things that we studied and were on previous exams that aren't going to be on here, and some of the things we didn't study. So just kind of point out some of the things that we didn't look at. So the digestive system, right? I don't like the way it's flickering. I think it's interesting. So here's some of the things that's not on the comprehensive exam. The digestive system, right? The functions of the digestive system, the 4 phases we talked about, digestion, absorption, motility, secretion, none of that's on there. The musculoskeletal system, the importance of bone tissue and homeostasis. We talked about calcium a lot, and fluxes in calcium and phosphate and how it's controlled and things like that. That's not on there. A couple things we talked about, a couple things we skipped. So the musculoskeletal system, we kind of compare and contrasted the 3 major types of muscle. We looked at the molecular basis of contraction. We skipped that for the most part. We skipped sliding filament and excitation contraction coupling. We skipped muscle mechanics to some extent. We skipped the metabolism and ATP a little mostly, and the types of muscle twitches we talked a little bit about. But all that's not on there. For immune function, the lines of defense isn't on there. The role of the lymphatic system's not on there. Integumentary system's not on there. Nothing for the reproductive. So we talked about, especially the numbers of things. That wasn't on there. We didn't, the categories of endocrines not on there. And enzymes, remember we didn't do enzymes in really any detail. So that's not on there. So all those things you just don't have to study. Okay. So something to think about there. So let's pause and find some other stuff to look at. Here is the systems comparison chart I was talking about for the final. So it takes the 5 systems we spent a lot of detail on. We're looking at the nervous, the endocrine, the cardiovascular, the respiratory, and the renal system, and then looked at some of the different things that affected it, the transportation, blood itself, heart rate, blood pressure, adrenal glands, proteins, neurohormones, neurotransmitters, hormones themselves, things like that. So this is kind of a nice little chart. I'm not going to go over it, but just make sure you understand it's there, and it does link some of the nice things between each system and how it does. So when we talked about the control of mean arterial pressure as an example, we looked at the nervous and endocrine system and kind of how it's related to the cardiovascular system with the different things with the heart, their vessels or stuff, and the blood, and how's transportation and communication. So, you can kind of look at it and take a study it. It's not great. It's, like I said, it's a little superficial, but it does give you a nice overview of how some of the things are connected, and certainly would probably be worthwhile some of your time as you prepare for the exam. Finally, here are the learning objectives for the comprehensive final exam. We'll just kind of run through them really quick. I'm not going to give you much of a rundown of them. So you should understand action potentials, both real and theoretical, including the reference numbers and calculations. What I mean by the reference numbers is, what's the equilibrium potential of sodium, right, is plus 60 millivolts, or the resting membrane potential, or threshold potential, or peak of the action potential. We've talked about all that, and how to do some of those things in terms of those numbers. Make sure you understand the action potential itself, and the depolarization, repolarization, hyperpolarization things. Make sure you understand what amphipathy is. And remember, amphipathy was when a molecule has both polar and non-polar ends, and its importance to the cell membrane, right? So the major portion of the cell membrane is a phospholipid bilayer, and it behaves the way it does because of the amphipathic nature of it. We talked a lot about ATP production, right? Very broad. How do we make ATP? You understand about glycolysis, activation, Krebs, beta oxidation, how we use proteins for fuel, things like that. You should understand the basic functions of each type of blood vessel. So what do arteries, arterioles, capillaries, venules, veins do, right? What's their basic function? Who stores the most blood? Which one controls blood pressure? Which one does exchange? Which one distributes blood? Which one has valves? Things like that. Be familiar with the numbers of things. We've had a ton of questions on numbers of things, right? How many structural types of neurons are there? How many functional types of neurons? How many ATP do you get from this or that? So make sure you understand those things. Number 6 is beta oxidation, right? What's the purposes of beta oxidation? What are the substrates? So we take a fatty acid, break it apart, take not a fatty acid. We eventually use the fatty acid, but we have to break that apart, the triglyceride first. So you take the triglyceride and break it apart with a lipase and that enzyme, and then take the fatty acid, which is an even number of carbons, and chop off the 2 carbon units. Remember, that was the beta bond, and make energy that way. So you be able to talk about that and how much energy we get from a triglyceride, which is 400 or 500 ATP, versus a glucose, which is 40 or less, depending on how we break it down. But the requirement for beta oxidation, remember, is most of that has to occur in the mitochondria. So to break down fat, we need oxygen, whereas we don't need that requirement for glucose, as an example. To understand the different bond types, so we're responsible for knowing the major bonds. So the ionic bond, the covalent bond, both polar and non-polar, and the hydrogen bonds, and how they work and things like that. You should understand the cardiac cycle, right? We saw a question like that on one of the exams previously. And so cardiac cycle, the 5 phases and how we do that, what cells are open, what valves are open, what's the pressure differences, and things like that. Cardiac muscle characteristics. So we did talk about a little bit about cardiac muscle and how it contracts and things like that, and some of the important parts of the cardiac muscle. We talked a little bit about the nodal tissue and things like that. You should understand the cardiovascular system anatomy a little bit, the heart and the chambers and how things like that, and the vessels and the blood itself. So nice overview. Cell membrane anatomy. So what's the properties of the cell membrane, and what's the anatomy of it? So we have pairs of things. We have 2 lipids, 2 proteins, 2 carbohydrates that are there, and what they do and what's their functional significance. So what's the cholesterol there for? What's the phospholipids there for? What's the integral proteins? What's the peripheral proteins? What's the glycolipids, the glycoproteins there for? We should understand cellular transport. So all the different active and transport mechanisms that we talked about. So in class, off the top of my head, we talked about the passive transport mechanism, simple diffusion, diffusion across a membrane, diffusion through a channel, facilitated diffusion, special case of diffusion of water across a semipermeable membrane, and osmosis. And then the active transport mechanisms that we talked about, primary active transport, secondary active transport, endocytosis, exocytosis, were all examples of those things. The different communication methods of the body. So remember, there were 3 communication methods. There were direct cytoplasmic transfer. We had local diffusion, and then we had the long distance communication, like an action potential or a hormone. So you should understand that. You should understand the conduction system of the heart. How does the heart conduction system work? And the SA node to the atria, to the AV node, to the bundle of His, to the left and right bundle branches, to the Purkinje fibers, and how all of that works. You should understand about the control of glomerular filtration rate. So how do we control GFR, and what are the ways we intrinsically and extrinsically homeostatically control GFR? We talked about those things in the last section. You should understand the definitions and determinants of plasma and formed elements and serum, and what's the difference, and what are the things we see in plasma and water and proteins and ions and other energy type things. What are the major formed elements, red blood cells versus white blood cells versus platelets, and all the subclasses of white blood cells. Determinants of capillary blood flow, so what allows blood to go through a capillary. We talked about that a little bit, not nearly as much detail as most of the other things, but a little bit. You should understand the determinants and voltages of resting membrane potential, and the ax potential, threshold potential, equilibrium potential. I don't see the ax potential versus the local potentials, but you should understand that. So what happens when you open a sodium, a potassium, a calcium, a chloride channel? What happens when you close them? Things like that. We had a little homework project on differences. You never turned it in, but you're asked to do it for the exam preparation. Differences and similarities between normal athletes and normal people, athletes, and cardiovascular diseased patients, and who would have bradycardia or tachycardia, or big stroke volumes or small stroke volumes, or things like that. 21 is crossed out because we didn't talk about enzymes, so that's not on there in any detail, at least. You should understand the iso, hypo, hypertonic, osmotic, and how to determine them. So if we put a red blood cell in a solution that was 360 milliosmoles, right, then what would happen to the cell? Well, 360 osmoles, remember the cell's 280, would pull water out. The cell would shrink. Cells shrink in hypertonic solutions, as an example. 23, what's the effect of the sympathetic and parasympathetic stimulation on the respiratory, renal, and cardiovascular function? So, if you stimulate the parasympathetic, what's going to happen to, your urine output, or, the heart rate and things like that? And sort of a trick question, because if we increase parasympathetic to the renal system, we can't, because there's no fibers that go there. Remember, it's sympathetic only. So things like that. The effect and causes of resistance in the cardiovascular system. So that would be Poiseuille's law, right? And big R, right, equals 8L times N over small r to the fourth. What's the relation between length, viscosity, and the radius, which is raised to the fourth power for Poiseuille's law? So you should understand that. You should understand a little bit about the electron transport chain, the general purpose, the end products, the substrates, the locations. So how do we take NADH and FADH2 and turn them into ATP? And remember, what we call that the chemiosmotic theory, and things like that, located in the mitochondria. Number 26 crossed out, right? That was on enzymes as well. Factors that affect an interaction of a protein in the binding site. So that's something we've talked about every section, right? And that's specificity, competition, saturation, and things that go with that. Famous people, so you'd expect to be able to see a famous person or two on the exam as part of a multiple choice question. So, who discovered homeostasis? Who's responsible for telling us that the concentration the partial pressure of a liquid will equilibrate to that of a surrounding gas. By the way, that's Henry. Right underneath that, another famous person, Fick's law, right? So Fick's law of diffusion is application. So we talked about that back in the first section. So remember, that was the flux, the rate at which a molecule moves across a membrane, right? So they have to be diffusible in the membrane. So it has to be small and non-polar, right? And that's proportional to the surface area times the concentration gradient divided by the membrane thickness times the membrane resistance. You should understand the fluid compartments, including the discrete and relative sizes. So the interstitial versus the plasma versus the transcellular fluid versus and that's all the extracellular fluid compartment versus the intracellular fluid compartment, right? And that's 2/3 in the intracellular, 1/3 in the extracellular, and all the things that go with those. And then what influences the movement of water between those, including osmosity and tonicity, which we kind of mentioned before. Let's talk about the 4 basic themes in physiology, right? So the 4 basic themes looked at biological energy, right? Compartmentation and structure function, right? So one part of that was compartmentation, homeostasis, and molecular interaction. No, that was part of structure function too. So doing all this off the top of my head. So 4 basic functions: biological energy, structure function relationships, homeostasis, energy, right? So those are the ones you need to worry about. The 4 classes, and if you haven't noticed yet, this is not in any particular order except alphabetical based on what the first word is, right? So the 4 classes of large biomolecules, so carbohydrates, lipids, proteins, nucleic acids, and the basic characteristics, we talked about that. The 4 major tissue groups in the body and their function, we didn't talk about a whole lot about that, but just a little bit. So epithelial versus connective versus muscular versus nervous. The gas laws that we saw in the last section, how to calculate them in the application. So not just Dalton's law or Henry's law or Boyle's law, but that's a good start. General traits and aspects of metabolic pathways. So how do we make energy and negative feedback and how they controlled and some of those things there. Particularly one of those would be glycolysis. So what's the substrate, what's the end product, what's the location, right? So the substrate is always glucose or some sort of sugar similar. The end product of glycolysis itself is always pyruvate. If it was anaerobic glycolysis, it would be lactate. It occurs in the cytosol. How many net ATP and total ATP do we get? And what happens to the NADH we make in that process that we've talked about before as well? Understand the difference between graded potentials and action potentials. And remember, they really don't have very many similarities except that it's movement of ions through some sort of channel or not moving ions through some sort of channel, to be honest. The big one thing, if you learn one thing this semester, that would be homeostasis, right? And so homeostasis, negative feedback, positive feedback, definitions and examples, that we've talked about that. Lots of terms for the next one. So how to define and calculate cardiac output. So you should know that equation: stroke volume, heart rate, ejection fraction, EDV, ESV, TPR, MAP, renal blood flow, GFR, respiratory rate, total volume, minute ventilation, alveolar ventilation, ventilation perfusion ratio. So that covers, the last 2 sections to a large extent. Pretty much, not all, but many of the calculations we're responsible for. And we saw kind of how they're related even in this last exam. Hydrophilic versus hydrophobic properties of water. So what's polar, what's non-polar, what dissolves in water, what doesn't, what needs a carrier, what doesn't, things like that we've talked about before. Reflexes of the cardiovascular system. The Frank Starling law of the heart is a reflex, really, as is preload too. So those are things to worry about. What influences the axe potential? So if we increase the temperature, what's going to happen to it? What's going to happen if we increase the diameter of the axon? What happens to the myelination in terms of how fast the axe potential goes, or the resistance? Should understand the different effects on the heart for inodromo and chronotropic, right? So inotropic is strength, dromotropic is conduction velocity, chronotropic is heart rate, and what positive and negative is, and how we can influence those with the sympathetic or parasympathetic system or some hormone. The intrinsic and extrinsic regulation of heart function. So how do we make the heart go faster or stronger, and what are the different things? So Frank Starling, sympathetic, parasympathetic, preload, afterload, venous return, calcium conductance, and all those different things. The structural and functional types and locations of neurons. So as an example, we only see this neuron in special senses, as an example. That would be the bipolar neuron, right? Things like that. Understand Krebs cycle. So again, what's the purpose? What's the substrates? What's the end product? What's the locations? What's my ATP yield? One turn of the Krebs cycle gives me 12 ATP total, right? It gives me 3 NADH, 1 FADH2, 1 ATP, 2 CO2s. And so understanding those type of things is what we're looking for. Lipophilic versus lipophobic substances. So lipophobic, lipid-fearing, lipophilic, lipid-loving, and that kind of relates back to hydrophilic, hydrophobic, but how do they behave? Long-term and short-term regulators of blood pressure. Remember, the long-term regular blood pressure is blood volume, more pressure. And the short-term regulator blood pressure primarily is the baroreceptor reflex, right? So what else we got? The second page, the major constituents of a cell. So the membrane, the organelles, the cytoplasm, the inclusions, and why they're there. So what organelles are important? What are mitochondria, endoplasmic reticulum, ribosomes, vaults, cytoskeleton elements, all those different things. What is important about those? The major steps to complete oxidation of glucose. So that means breaking glucose down completely to get energy. So starting with glycolysis, then going through activation and Krebs. The oxidative processes, including aerobic respiration, and then contrasting with anaerobic. So, we can have multiple fuels, substrates, whether it's glucose or some sort of fat through beta oxidation, where in the anaerobic metabolism, we can only use glucose, and, contrasting those things. The partial pressures of oxygen and CO2 in the various parts of the body. So what is it in the lungs or the tissue and things like that. Going way back to the beginning of the semester, the pH scale, how it works, what's an acid, a base, a salt, right? It's a log scale, and if we went from 5 to 3, we became 100 times more acidic, as an example. The 5 phases of respiration, right? So how do we get air from the outside world to the inside world? And that was literally a question that most people seem to do on the last exam. So understand that. Physical quantities and definitions of matter, energy, mass, things like that. How do we specially calculate what matter and mass is and things? Not in a lot of detail like we did in lab, but in a little bit of detail. What's the solution versus a suspension, and what's dissolved in osmolarity and things that kind of include with that. Number 57, processes that lead to urine production, right? That was another question on the lecture exam that some people did. And so filtration, reabsorption, secretion, and what that includes. Properties of atoms and molecules, and what's an isotope, an ion, and radioactivity, and things like that. So some of that basic chemistry stuff that we didn't really talk about in class, a little bit, but most of it was online posted stuff that you're still responsible for. Properties of red blood cells and measurements and production and hemoglobin. So, hematopoiesis and especially erythropoiesis with EPO and how that works. And then talking about the various things we measure with red blood cells, the total count, the hemoglobin, the hematocrit, and things like that. And where's oxyhemoglobin and deoxy and, carbamino and carboxyhemoglobin and all those different things. Properties of water, what makes water so special. It's a great heat sink. It's, important in terms of biological function. It helps to stabilize temperature, which sort of relates to the heat sink. It allows an aqueous environment for reactions to take place. A whole bunch of different things. What's the structure and function of proteins? How they work as enzymes, and they're very different in terms of by far the most biologically diverse type of biomolecule. And, the primary structure and the denaturation and things like that. We'll talk a little about reactive and active hyperemia in the cardiovascular. Remember, hyperemia is an increase in blood flow. And so active is because of metabolism, and reactive is following occlusion. Receptor turnover, so downregulation, upregulation, what causes those things. The relationship between blood pressure, blood flow, and resistance. So those hemodynamics that we talked about. So remember, LMNO-PQR. So P, pressure, equals flow times resistance, as an example. And then being able to figure out the other ones independently by rearranging the equation. Starling's law of the capillaries and the calculation of net filtration pressure. So we did that one at the end. Subatomic particles, including charge and location. So where are electrons located primarily, and what charge do they carry, and things like that. The theory of mechanism versus teleology, literally the first day of class. Remember, mechanism was a scientific way of doing things, and teleology was the belief that some other thing is affecting natural phenomenon, or attributing a human purpose to things that aren't human is also another form of teleology. And then the major afferent fibers for the somatic and autonomic nervous system. So what do they look like? What are the neurotransmitters? What do they do? And things like that. So that's the end of this, and probably a good place to cut this off. But that's kind of a quick overview, and some of those things are a little more important than the other, and I tried to point out the ones that tend to be a little more important and less important. But study hard and keep with it, and do the things that have made you successful up to this point, and pay attention to these things that we just looked at. Pay attention to the group problems. Pay attention to the notes, and think about what type of questions would be there. And again, just to quickly review the exam, there are 60 questions that are multiple choice that cover primarily the first 3 sections. The take-home essay portion is 5 questions worth 8 points apiece, as opposed to fewer that we've had recently, but more points. So the first question tries to encompass some things that we worried about in class this semester, and tie some things together, especially mathematically. The second question tries to take a fairly central theme of the class and connect some concepts, and that one's important. And in neither one of those do you get a choice. There's just one question that you answer. And then there are 3 sets of 4 questions each that you get to just choose one from each set. And I tried to group them. One set I think is pretty easy with some basic information that's just kind of regurgitating it. One of them is asking you to do a little more thinking and thought stuff and go ahead and like extrapolate some information. And the last set is trying to ask you to do some math. So just, you get one from each set, and the two that you don't have a choice from, and that would be 5 questions total for that, which would be 8 points apiece for a total of 40 points. So the way you've done things before, you've been successful, do them again. And if not, in this short amount of time you have left, prepare the best you can. So good luck on Wednesday, and hopefully you meet your goals.